The next segment of this series on critical care and genomics is to do with precision medicine and genomics. And I guess it's what we all aim to do, is to provide that most precise and personalized medicine to a particular patient. And this is just a quick snapshot of where genomics comes into the picture. What is precision medicine? Well, it's the application of modern genetic technology. It takes along into account the living environment, including the clinical data of the patient. It puts it into context. You use other things such as molecular imaging techniques, and most importantly, increasingly over the years, is the bioinformatics technology. What are we aiming for? We're aiming for a more accurate diagnosis and a much improved treatment. And also, of course, to look ahead in a way to try and prevent disease, and on a more of a public health way. Now, dealing with precision medicine is a bit like looking at the stars, is that it is so vast, and this is a picture up in the Warren Bungall's conservatory, that if you're a stargazer or an astronomer and you look at the stars, where do you start? Which star do you start with? Which stellar system? And of course, the trouble with genomics and precision medicine is exactly the same. It is huge. So let's go back to basics, and we've seen this before, but in fact, way back in 1988, yes, it's a long way back, there was a study that came out of the New England Journal that looked at the epidemiology of genes and sepsis, and this was before we could do all the fancy things we could do today. And the important thing here is that if your biological parents died of infection, it didn't matter whether you lived with them or not, or you're raised with another family, you were nearly 6 times as likely also to die from a septic cause. So what we're doing with precision medicine and genomics is to try to take the genotype to the phenotype. And basically what we're doing and what has happened with that is in the 1990s, over the last few years, particularly in the 1990s, it was the Human Genome project that really brought all this together. And of course, since that time, we've advanced even further with next generation sequencing, supercomputing. We now have ways of dealing with huge amounts of bioinformatic data. And of course, in the post-genomic era, which means after the success of the Human Genome project, we're now looking at other multiple levels of investigation, such as the epigenome, the transcriptome, the proteome, the microbiome comes into it, and the metabolome. And really what we're looking at is this line down here from genomics all the way down to the phenotype. We must keep the individual in mind, because whatever we do as doctors, we're aiming to give the best treatment we can to a particular individual. Just to go back on some of the terms that we've used, and you've come across this before, but to remind ourselves, if we're going to look at the DNA, we can deal with the whole genome sequencing, which means we can look at all the genes in a particular person, or we can just look at those genes that code for proteins, the exome. And so we can have a whole WES, which is whole exome sequencing. Or you can take it even one step further and say, I just want to look at 2 or 3 or 4 or 5 or 10 or 20 specific genes and have a gene panel. This is far too much on it for you to see, but let's just take a little section out of it. This is oncology, and just one little part of precision medicine, genomics and oncology. And what we're looking at here is to say that if you have a particular gene, and I've chosen BRCA, because BRCA is actually pretty well known to most doctors. We don't normally deal with it in our practice, but we're aware that if you have a breast cancer or ovarian cancer, that in fact you may have a family history of that, and you'll find that in the family is this particular gene called BRCA. We actually do it on DNA testing on tissue from the body, and we look at various mutations of the BRCA gene. Why is it important? Because armed with that information, we can choose highly selective and very expensive anti-cancer agents. So this is just one example of how you use genomics to hone in on providing that particular person with a state-of-the-art treatment. Now, another example, and this is a little more complicated, but just keep it simple insofar as that what we're looking at is people who are prone to infections from a genetic cause. And on this side of the diagram is people with an intracellular infection, and if you have defects in these particular proteins, then in fact you're highly susceptible to having infections. This is an extracellular infection, an extracellular antigen, and if you have abnormalities in these particular genes, then in fact you're more highly likely to have infections. So once again, if we go back to that study in the New England Journal in 1988, a number of these people would have had this sort of defect in their inflammatory system, hence the high susceptibility to dying from sepsis. Now, the interesting thing is that when we look at genotype, we're really talking about genomics, and the DNA, the genome itself. And that's reasonably straightforward. We see a link between the gene, the protein, and the outcome for that particular person. And the important thing to take this even further is, in fact, what if we can edit the gene? If we can go in and cut out a bit of the so-called bad gene and put in a new gene in its place, can we actually cure that person's underlying problem? And of course the classical one would be something like a cystic fibrosis, or even insulin would come into it. And it's a good opportunity here to just introduce the word CRISPR, because it's kind of fascinating, and if you do genomics then you'll understand why this word has become so important, and in fact why two scientists, in fact two women in the 2020 obtained the Nobel Prize for working on this. And it's being explored in research on a wide variety of diseases, particularly single gene disorders, cystic fibrosis, haemophilia, and sickle cell disease. And the whole idea of CRISPR is to in fact go in, take out the bad gene, and can you edit the gene by not only taking it out but putting a new one in. Now the word CRISPR actually stands for clustered or regularly spaced short palindromic repeats, which is why no one uses the term, it's too complicated. And the other thing you should be aware of is this CRISPR and the enzymes that go with it. I've just chosen Cas9, or the CRISPR associated protein 9, and these are very important in the process. Just to give you a pictorial view of it, now this is fascinating because this was determined in bacteria. Now bacteria of course have faced virus infections forever, since they've been around, and so they've developed ways of fighting the virus. And what they do is this, on the this part of the diagram here, you've got the DNA, which is the virus you can see here. It puts the DNA, the phage DNA goes into the bacteria. What happens is that in fact the bacteria goes in and cuts out a piece of that DNA. Then it takes that DNA and incorporates it into the bacterial DNA. Interesting. And what they do is they do it at regular intervals, which is where the word CRISP comes from, is that you have repeated sequences in the DNA. Why? Because later on when a virus comes with the same DNA or very similar DNA, and the DNA gets into the cell, the DNA in the bacteria has this information, and as a result it actually activates the Cas9 gene to go in and actually slice up the phage DNA and therefore stop the phage from replicating inside the cell. And of course the next step is to say, if the bacteria can do this, can we apply this technology to humans? So gene editing is a fascinating area. It's only beginning. We have no idea where it's going to go, but I would think over the years we're going to see some rather, I'd like to think we're going to see some spectacular results from it. Now, the interesting thing though is gene editing means we're always talking DNA, because DNA is a very stable structure, and so it's easy to, I won't say it's easy, let's say it has the advantage of not changing very much, so you can relate the gene to the DNA. But what happens with the rest of the genomic expression when you go from genomics, transcriptomics, proteomics, metabolomics, and down to the phenotype. Now, this is where we're very interested in critical care medicine, because this is where we see opportunities as to how we can manipulate a person's illness and thereby improve the outcome for that particular person. And of course, we've already had a presentation in this series on transcriptomics and the outcome in sepsis. And I'd just like to mention this one. It's a fairly well-known one. It's the classification of patients with sepsis according to blood genomics endotype, which means that when somebody has a particular illness, you measure what RNA is being produced. And therefore, as a result of this, you can come up with particular patterns within a person. And this was in the Lancet Respiratory Medicine 2017 by European and some American groups. Now, this is the result of this particular paper, where in fact if you take a whole lot of gene expression, transcriptomics, you can divide it up into outcome, and therefore if you can determine how these groups differ in the gene expression, then maybe you can go in and alter that or be more precise with the treatment you give that patient. We've already presented some of our own work in this regard, and there's a big step, and we know this from our own research activity, to go from large numbers of expressed genes to finding genes, narrowing it down so you can actually manipulate the patient's immune response. Now, the fascinating thing is, of course, is that it just becomes a little more complicated, because as we've discussed before, only 1% of your DNA ends up translating to protein, and 99% does not. So what does this other 99% do? We've seen this slide before, where we know now that these other DNA, RNA expressions can alter the pathway of other expressions that go from DNA down to protein, either modifying the protein, interfering with the protein, stopping the protein being produced. And just for the final part of my talk, I'd like to move on to an indication of how this is coming into critical care medicine. I'm going to talk about microRNA. Now, as we mentioned, the non-coding RNA can be divided into three major areas: the microRNAs, the long non-coding RNAs, and the circular DNAs. And I'm not going to go through all this diagram in depth to say the DNA produces a microRNA that comes out into the cytoplasm. But what happens, it goes into this particular thing here called RISC. And the RISC is RNA-induced silencing complex. And so the RISC, if produced, will actually interfere with the expression of other genes. So you can see how this could influence the outcome in sepsis or other critical care conditions by having RNA that will interfere with the expression of other genes. This is just a diagram pointing out that we're looking at the heart here, because I'm just going to touch upon the heart briefly to say that the non-coding genome generates a vast number of these non-coding RNAs, as mentioned, micro, long non-coding, and circular RNAs. And this is just one to do with the heart, that in fact now they've looked at the microRNAs, and you can find these microRNAs that are either upregulated, as seen in green, or downregulated, seen in red, which are associated with myocardial infarction, hypertension, contractile defects, arrhythmias, and inherited cardiomyopathies. I've just made the contractile defects a little bigger because this is what we're interested in when you get sepsis-induced left ventricular dysfunction, when you get Takotsubo syndrome, when you get anything that interferes with cardiac function as a result of something else going on to the body. We know now that these microRNAs are playing a role. So if we could just have a little understanding of how they are affecting the working of the heart, maybe we can improve the outcome of that patient by improving cardiac and cardiovascular function. And just to finish off, the interesting thing is, of course, we see the genome is working through other parts of the system, the non-coding RNA affecting other coding RNAs, etc., etc. But now we actually have evidence that non-coding RNA can act directly on pathogens itself, and just not regulate the production of proteins. And this is just a paper published earlier this year, where you've got non-coding RNA will interfere directly with the influenza virus replication. And so this goes back to the Crispr idea that the bacteria has, that if you can actually have genetic material in your own genome that in fact doesn't go through the transcription translation protein, but goes directly to interfere with the pathogen, this opens up a whole new vista of possible treatment for these patients. Now, in summary, we've just barely touched the surface, and there's huge areas we haven't gone anywhere near, such as pharmacogenetics being an example. The field of precision medicine and genomics is huge. We have to research not only the DNA but also RNA and other aspects. The important thing is that as a critical care physician, you might try and avoid all of this because it's all too hard, but the reality is our future management, or the management of future critically ill patients, will be based on today's research. So it's useful to at least have an idea of what people are talking about when they use certain terms. And so really, clinicians need to develop a basic comprehension of the subject. Not detailed, just enough to understand. So in 10 years' time and 15 years' time, you don't find that you've really been pushed to the side of things because people are using terms and terminology for which you don't have any of which you have no understanding. And on that, I thank you.